Okay, let's just jump in. Picture this. What if you could step outside, soak up some light, and get energy from it? Like a plant.
Uh-huh.
I mean, human photosynthesis, it sounds like the wildest science fiction.
Absolutely.
Imagine the possibilities. Maybe needing less food, having more consistent energy throughout the day.
It absolutely sounds like something out of a futuristic novel.
Hmm.
But this concept is actually moving beyond pure speculation and into areas where scientists are actively, well, exploring related ideas.
Exactly, and that's what we're diving into today. We've got this incredibly intriguing source material, a research analysis paper specifically titled Human Photosynthesis and Optimal Cellular Nutrition, a comprehensive research analysis.
Yeah, and what makes this paper so fascinating is that it takes a genuine, recent scientific breakthrough, putting parts of plants into animal cells, and connects it with some really radical, I'd say even controversial ideas.
Right.
It leans heavily into the framework developed by Dr. Jack Kruse, often referred to as quantum biology.
Right. It's this blend of solid lab work and a theoretical framework that really pushes the boundaries of conventional biology. Our mission today is to unpack this paper for you, understand the core concepts it presents, figure out where the established science ends and the speculation, begins,
Yeah.
and then explore what all of this could potentially mean for human health, energy levels, and performance, according to the analysis provided.
Get ready for some genuine aha moments, I think.
Definitely, and probably a few things that'll make you stop and think, maybe scratch your head a bit.
For sure. And this isn't just abstract science either. It touches on some very fundamental questions about where our bodies truly get energy, how light might be influencing us in ways most of us don't even consider,
That's a key point.
and what optimal nutrition might look like through this entirely different lens. It makes you reconsider basics you thought you already knew.
Absolutely. So let's unpack this paper, starting with that key scientific breakthrough. It points to as potentially opening the door to this whole idea of human photosynthesis.
Okay, so the core premise the paper starts with is that the concept of human photosynthesis is potentially shifting from pure science fiction towards something, well, tangible.
And the specific event driving this idea, according to the paper, is this significant scientific achievement from 2024 by a team of Japanese researchers, Matsunaga and colleagues. What did they actually do in the lab?
Well, they successfully integrated chloroplasts, those tiny structures inside plant cells that capture light for energy.
Right, the photosynthesis engines.
Exactly. Specifically from red algae, and they put them into mammalian cells. The cells they used were Chinese hamster ovary cells, often called CHO cells.
CHO cells, okay. A common type used in lab research.
Very common, yeah. A workhorse in biotech labs.
And did these cells actually do something with the chloroplasts once they were inside?
Yes. Remarkably, they observed photosynthetic electron transport within these cells. That's the initial step, really, of photosynthesis, where light energy is captured and transferred.
Okay.
They even detected oxygen production, which is a key byproduct of photosynthesis.
Wow, so actual photosynthetic activity.
Some level of it, yes. The study found that a small percentage, somewhere between 1 and 20% of the CHO cells, successfully took up and showed activity from these chloroplasts.
Okay, so the paper interprets this achievement as a paradigm shift. The idea being that this could potentially allow mammalian cells, and theoretically, eventually human cells, to perform light-dependent photosynthesis alongside their traditional way of making energy, which is mitochondrial respiration.
Right, supplementing ATP production.
Right, supplementing ATP, that's the basic energy currency our cells use, and providing necessary reducing equivalents. What are those exactly again?
Basically, they're like electron carriers crucial for many cell processes. Think of them as charged batteries for chemical reactions.
Got it. So the big concept here is cells potentially becoming more energetically self-sufficient using light.
That is exactly how the paper frames this achievement. Now, here's where the source evaluation provides some crucial context.
Right.
The Matsunaga study is a real, genuine scientific achievement. It's important work. It opens new avenues for researching these cellular biohybrid systems.
Right.
But interpreting the single study as proof that widespread, functional human photosynthesis is imminent in a complex human body is a significant leap.
Right.
Human photosynthesis, in that broad sci-fi sense, is still very much conceptual, not a currently recognized biological function in humans.
So the study is real, solid science, but the interpretation that this means human photosynthesis is just around the corner. maybe pumping the brakes a bit.
Exactly. The source analysis highlights critical limitations right from the Matsunaga study itself. For one, the chloroplast activity they observed was transient.
Transient, meaning?
It only lasted for about 48 hours. That's a very short lifespan for something meant to be a sustained energy system, you know?
Yeah, not exactly long-term integration.
Not at all. Second, as you mentioned, the integration was limited. Only that small percentage of cells, 1 to 20%, took up the chloroplast successfully.
And importantly, they were CHO cells, lab cells, not complex human tissues in a living body.
Precisely. And these results were in vitro, meaning in a lab dish, translating something seen in isolated cells in a controlled environment to the complexity of a living human organism.
Huge jump.
With immune responses, different tissue types, metabolic demands. It's a monumental challenge.
Okay.
And the source analysis also points to a very specific detail in the Matsunaga paper. It noted the observed increase in cell growth rate was, and I quote, likely due to increased nutrition concentration rather than a photosynthetic endosymbiotic relationship, as photosynthesis could not occur without illumination.
Oh, okay. That little phrase is key, without elimination.
It is. It hints that perhaps the benefit wasn't just from photosynthesis providing energy, but possibly from nutrients leaking out of the chloroplasts as they were integrated, especially when light wasn't present, like a little nutrient pack.
Wow, that's a subtle but really critical distinction. So the takeaway from this section, based on our source evaluation, is successfully integrating chloroplasts into some lab cells was a real and exciting step in cellular engineering.
Definitely.
But functional human photosynthesis as a biological reality is still very far off, if it's even possible with this specific approach.
That's the balanced view. It's a proof of concept for cellular engineering, but it doesn't confirm functional human photosynthesis, as the paper's title might perhaps imply.
Right. Which brings us to the other major component of this paper, the framework developed by Dr. Jack Kruse. This is where things get into some pretty non-conventional biological ideas, wouldn't you say?
Definitely. This paper heavily integrates Dr. Jack Kruse's quantum biology framework, which offers a radically different perspective on the fundamental drivers of life, health, and energy.
A framework that proposes light is far more important than traditional biology typically acknowledges.
That's putting it mildly, yes.
So what are the core tenets of this framework as presented in the sources? Where does it start?
At its absolute core, Kruse's framework posits that light is fundamental. It's presented as the primary organizer of life and the foundation of the entire food web.
Wow, primary organizer.
Yeah. In this view, food itself is almost secondary. It's described abstractly as a story about how electrons are managed.
So energy isn't just about calories from food. It's more about how we capture and manage electrons. And light is the key player in that whole process.
That's the central idea. He links our mitochondria, the cell's powerhouses, directly to light. He emphasizes that DHA,
The omega-3 fatty acid.
that key omega-3 fatty acid, yes, found in things like fatty fish. He says it's crucial for converting light into usable electrical current within our cells.
Okay.
And he highlights a specific type of light, blue light, particularly from artificial sources like screens, LEDs.
The kind we're bathed in all day.
Exactly. He sees that as being destructive to DHA and thus impairing this light-to-energy conversion process.
So according to this framework, looking at your phone at night isn't just messing with your sleep-wake cycle, your melatonin.
Which it does.
Right. But it's also potentially directly damaging the cellular machinery needed to turn light into biological energy.
That's the assertion made within this framework, yes. He also incorporates ideas from researchers like Gerald Pollack about the fourth phase of water, or exclusion zone water, EZ water.
Right, structured water.
It's a special structured layer of water that forms next to cell surfaces, and Kruse links its formation and stability to cellular energy production, emphasizing light's essential role in creating and maintaining this structured water.
Okay, so light is foundational. It links to mitochondria via DHA. Blue light is disruptive, and water structure matters. And then there's this idea called the light transmutation theory. That sounds pretty out there.
This is indeed a central and I'd say highly controversial concept within Kruse's framework. It suggests that light can directly create essential vitamins, minerals, and biochemicals in the body.
Directly create them, not just trigger a process.
The theory suggests a more direct conversion of light energy into molecular structures, potentially bypassing or significantly altering traditional metabolic pathways.
And the main example he uses is vitamin D3. But we know sunlight is involved in making vitamin D. That's standard biology.
Right. The established science is that sunlight initiates vitamin D synthesis in our skin. Specifically, UVB light hits a cholesterol-like molecule called 7-DHC, converting it into pre-vitamin D3, which then follows various metabolic steps in the liver and kidneys.
Okay.
Kruse emphasizes the quantum nature of this process and argues for the superiority of sunlight-derived vitamin D3 over supplements. He uses this analogy. Taking supplemental vitamin D3 is like trying to hire someone else to do your push-ups for you.
Ah, okay, I get the analogy.
But he adds a twist. He describes this process as involving light-changing cholesterol to sulfated cholesterol and then to sulfated vitamin D3, suggesting light is somehow directly involved in adding those sulfate groups.
So he's taking the known process of vitamin D synthesis and reinterpreting the mechanism through this quantum lens, suggesting light does more than just initiate.
Partially, yes.
Yeah.
He applies concepts from physics, like Einstein's photoelectric effect, where light hitting a material can eject electrons, to biology. He states, a cell is a quantum measuring device for light's frequency.
Okay.
He suggests proteins, especially those with certain structures like aromatic rings, act as light capture mechanisms. And he talks about tiny structures or even single atoms becoming programmable quantum nanomachines for light photons to convert light energy directly into molecular structures.
Quantum nanomachines.
Yeah.
That really does push the boundaries of current understanding, and he sees structures like porphyrins found in chlorophyll and our own hemoglobin as crucial biological solar panels.
Yes, within his framework, these are identified as key structures that capture photons and convert them to usable biochemical energy or compounds. The paper also mentions biophoton emission.
That's the weak light cells give off.
Exactly, ultra-weak light that cells naturally emit. He suggests it might indicate mitochondrial energy status and potentially be a subtle communication system between cells.
Okay, that's a lot to take in. Light is the core, linking to mitochondria and structured water, blue light as a disruptor, and this idea of light directly creating nutrients.
Yeah.
What does our sources' evaluation say about the mainstream scientific standing of these specific Kruse framework ideas? Where does this fit?
This is where the evaluation provides really essential perspective. It clearly states that Dr. Jack Kruse's theories, while definitely intriguing, are highly debated and largely lack mainstream scientific consensus and robust empirical support.
Okay.
This is especially true for the light transmutation theory that suggests light directly creates biochemicals in ways that bypass or significantly alter established metabolic pathways.
So while he uses terms and points to phenomena that exist in science, like, yes, vitamin D synthesis from sunlight happens. Yes, molecules absorb light. Yes, cells emit biophotons. His interpretations and extrapolations go significantly beyond what's broadly accepted.
Exactly. He draws on real concepts, but his conclusions, for example, that light directly adds sulfate groups to vitamin D3 as a quantum enhancement, or that light can bypass enzymatic processes to create other vitamins. These are significant departures from standard biochemistry and cell biology. They are not widely supported by evidence.
Right.
Similarly, while biophoton emission is a real area of research, its proposed role as a primary energy source or a sophisticated communication system in humans, as described in his framework, is considered speculative and not accepted in the mainstream scientific community.
Got it. So the paper uses this framework extensively, but it's important for you, the listener, to understand that much of that framework, particularly the more quantum aspects like direct transmutation, is considered outside established science by many researchers.
That's a fair summary.
Now, how does the paper try to connect that initial chloroplast breakthrough with these Kruse ideas? Does it draw a direct line?
Well, the paper attempts to bridge the two concepts. It suggests that ideas from the Kruse framework, like the importance of structured water, easy water, might somehow facilitate the integration of chloroplasts into animal cells.
Okay.
Or perhaps that existing biophoton capabilities within cells could be enhanced by this integration.
But does the Matsunaga study itself actually provide evidence for those specific links? Do they measure structured water or biophotons in relation to the chloroplasts?
No. The source evaluation points out that this connection made in the paper between the results of the Matsunaga study and Kruse's specific ideas about structured water or biophotons is speculative.
Ah.
The Matsunaga research findings themselves did not directly investigate or support those particular links within the Kruse framework.
Okay, so the scientific validation for that specific connection isn't there in the study itself. It's more of a theoretical overlay by the paper's authors.
Correct.
However, The theoretical implications, if a system combining our biology with photosynthetic elements were somehow successful way down the line, they are pretty astounding. What does the paper suggest could be the outcomes?
The theoretical implications, based purely on the concept, if these massive hurdles were overcome, are indeed profound. The paper suggests a hybrid energy system. Our mitochondria plus some form of light harvesting could lead to significantly enhanced cellular energy.
More energy.
This means potentially increased ATP production, less dependence on external glucose as a primary fuel, and maybe even internally generated oxygen.
More energy, less reliance on food, making your own oxygen.
Yeah.
That really does sound revolutionary.
It absolutely does. It also suggests potential metabolic boosts like increased NADPH, another crucial molecule involved in energy and balancing cellular chemistry.
Right.
Potential carbon fixation like plants do, though the scale of that is debatable, and greater metabolic flexibility, meaning cells could adapt better to different conditions.
Oh.
And drawing heavily on Kruse's more speculative transmutation idea, it implies that light could directly synthesize essential nutrients inside the body, things like vitamin T3, various B vitamins, activate minerals, and create beneficial sulfated compounds.
So the idea is your body could potentially make its own vitamins and other crucial molecules just from light exposure.
That's the theoretical possibility explored in the paper, building directly on Kruse's framework. And if that were possible, it could open doors for totally new therapeutic approaches.
Such as?
Potentially treating metabolic disorders, dramatically enhancing wound healing, improving tissue engineering, and maybe even playing a role in mitigating aging processes by enhancing cellular resilience and energy.
These implications, while clearly based on overcoming colossal scientific challenges and incorporating some speculative ideas, are just fascinating to consider. It really makes you think about the future of health and medicine in a way you probably never have before.
It pushes the boundaries of our current understanding of human biology and energy metabolism in some really dramatic ways.
Now, while the paper doesn't directly tie the chloroplast study to specific glands like the pineal and pituitary,
Right, it doesn't make that specific link.
it does rely heavily on Dr. Kruse's framework, which does have a lot to say about how light impacts these glands and overall human performance. Let's look at that part of the analysis derived from Kruse's ideas.
Right, exactly. So now we're shifting focus slightly to analyze how the Kruse framework, which is so central to this paper's broader arguments, discusses light's influence on specific endocrine glands and consequently human performance.
Okay, let's start with the pineal gland. Most people know it's connected to sleep because of melatonin production.
Exactly. The pineal gland is a key regulator of our circadian rhythms, primarily through its production of melatonin. And melatonin production is highly sensitive to light signals received via the eyes.
Light suppresses it, dark stimulates it.
Correct. Light, especially blue light, suppresses melatonin production while darkness stimulates it. That's well established.
And what's Kruse's perspective on this gland's connection to light? Does he add layers to that?
He does. Within his framework, artificial blue light is highlighted as being profoundly disruptive to the pineal gland and melatonin production. He links this disruption back to his idea about blue light causing destruction of DHA, which he posits impairs the cell's ability to handle light signals properly.
Ah, the DHA link again.
Yes. He argues that maintaining natural light cycles, getting sunlight during the day, avoiding blue light at night is crucial for optimal pineal function. He also sees melatonin as much more than just a sleep hormone, presenting it as a powerful antioxidant essential for mitochondrial health and autophagy.
Autophagy. Cellular cleaning, right?
Exactly, like cellular self-cleaning and recycling.
Yeah.
He describes melatonin production itself as a quantum process.
Okay.
The source notes that his framework, like some others in alternative health circles, also discusses potential pineal calcification, the buildup of calcium deposits, sometimes speculatively linked to environmental factors like fluoride or EMF, suggesting this impairs the gland's function.
So according to this view, hitting snooze after your phone alarm blasts blue light at you isn't just making you groggy, it's potentially interfering with fundamental cellular repair processes tied to melatonin. That's the implication.
That's the connection made within his framework, yes. The resulting impact on performance, from this perspective, includes significant sleep disturbances, hormonal imbalances, reduced antioxidant capacity, and potentially cognitive impairment over time.
What about the pituitary gland, often called the body's master gland? How does light fit in there according to Kruse? Is it also light sensitive?
Well, the pituitary gland itself isn't directly photosensitive like the pineal gland. However, it's strongly connected to light exposure via the hypothalamus, which does receive light signals from the eyes.
Okay, an indirect link.
Yes, an indirect but crucial link. This influences the release of many hormones, including cortisol, through a pathway called the HPA axis. That's the hypothalamic pituitary adrenal axis.
Right, the stress axis.
Exactly.
And Kruse's perspective on this pituitary connection.
His framework emphasizes that getting natural light exposure, particularly in the morning, is critical for setting a healthy diurnal cortisol rhythm that's the natural daily pattern of our main stress hormone.
The cortisol awakening response.
Precisely. It helps establish that necessary bump in cortisol that helps us feel alert and ready to go in the morning. Artificial light at night, he argues, disrupts this rhythm, potentially keeping nighttime cortisol levels higher than they should be, which can impact sleep and recovery.
Okay.
He also links appropriate light exposure to other performance-related hormones and brain chemicals, like dopamine, which affects mood and motivation, and leptin sensitivity, which is crucial for regulating appetite and metabolism. He asserts that blue light exposure at night specifically lowers dopamine levels.
Interesting.
His framework even mentions neuropsin, which is a type of photoreceptor found not just in the eyes, but also apparently in skin and other tissues. He suggests it could transmit light signals that impact systemic hormonal responses, potentially influencing the pituitary axis even without going through the eyes.
So the amount and type of light you get could impact your stress levels via cortisol, your mood and drive via dopamine, and even how your body handles appetite signals via leptin.
According to this perspective, absolutely. The performance impacts discussed here range from energy and alertness levels via cortisol to metabolism and weight regulation, leptin, dopamine, mood and motivation, dopamine, and overall stress resilience, the HPA axis. It paints a picture where light is a major regulator.
And beyond these specific glands, the paper, leaning on Kruse, talks about light impacting broader quantum functions at the cellular level that affect performance. What does that refer to?
Right. The framework proposes that light profoundly influences fundamental cellular processes that underpin overall performance beyond just hormone signaling. Mitochondrial efficiency, for example, is seen as highly dependent on light quality.
Natural versus artificial.
Exactly. Natural sunlight is presented as beneficial, enhancing efficiency, while artificial blue light is seen as damaging to mitochondrial function over time. Structured water, that EZ water we mentioned, is presented as being light-dependent and essential for both energy storage and optimal cellular function, like enabling protein folding and cell signaling. The cell's redox potential.
The balance of oxidation and reduction.
Yes. Essentially, it's a balance of electron borrowing and lending, which is crucial for energy production and antioxidant defense. This is also described as being maintained by natural light and disrupted by artificial light.
Okay.
And biophoton emission, while again speculative in this context, is viewed within the framework as a potential subtle communication system influenced by cellular energy status and light exposure.
So the big overarching idea from Kruse's framework, as highlighted in this paper, is that human performance isn't just about food and exercise. It's fundamentally driven by the environmental light environment.
That's the core message.
Optimize your exposure to natural light, minimize harmful artificial blue light at night, and you can profoundly enhance cellular function and overall performance. Is that a fair summary of his view?
That is the central tenet of his framework as presented. And again, it's crucial to circle back to the source evaluations perspective here.
Right. The reality check.
The reality check. While Kruse's ideas touch upon concepts rooted in established science, yes, light affects melatonin, circadian rhythms are real, DHA is important. Biophotons exist his specific framework, particularly the direct light transmutation concepts and the detailed quantum mechanisms he proposes for how light affects glands and overall performance, is largely outside mainstream scientific consensus. It lacks robust empirical validation in that specific context.
So incredibly fascinating, potentially paradigm-shifting ideas presented in the paper,
Yeah.
but originating from a framework that hasn't been broadly accepted or validated by the wider scientific community. That's the key takeaway here.
Precisely.
Which leads us neatly to the final section of the paper.
Yeah.
What are the major challenges to making any of this human photosynthesis concept a reality, even the basic chloroplast integration part? And where does future research realistically need to go?
Well, the paper itself identifies very significant hurdles. On the technical side, the transient nature of chloroplast activity, remember, lasting only 48 hours in the study, that's a huge problem for any practical application.
Right. Needs to be stable long term.
Absolutely. The low integration efficiency, only getting chloroplasts into 1 to 20% of cells, just isn't enough for widespread function in a tissue or organ.
Okay.
And a really fundamental physical limitation is light penetration depth in human tissues. Sunlight doesn't reach deep organs or muscles effectively. How do you power these chloroplasts if they're deep inside the body?
Good point. Like internal solar panels without sun.
Exactly. And then there's the massive challenge of long-term compatibility and stability of foreign chloroplasts within a living organism. Will they survive? Will they keep working?
And the biological hurdles. The
Yeah.
body isn't likely to just welcome foreign plant parts with open arms,
Yeah,
right?
you got it. A major hurdle is the human immune response. The body would likely recognize the chloroplasts as foreign invaders and mount an attack to destroy them.
Yeah, the immune system doing its job.
Precisely. There are also complex questions about the genetic stability of the chloroplast DNA within a host cell over time. Would it remain stable? Could it mutate? And the huge challenge of maintaining the overall metabolic balance of the cell and the entire organism with this added photosynthetic component. How does it integrate?
And, of course, looming over all of this are the crucial ethical and safety considerations.
Absolutely paramount. Any potential human application would involve elements of genetic modification, potentially altering the human germline depending on the approach, raising significant ethical questions that society would need to grapple with. Informed consent would be critical, ensuring people truly understand the risks and unknowns, and rigorous, extensive long-term safety testing would be absolutely essential before anything like this could even be considered for human trials, let alone widespread use.
Okay. Despite these immense challenges, the paper does map out potential future research directions. What are they suggesting scientists should work on?
Well, the suggested directions logically align with tackling the hurdles we just discussed. Research into optimizing chloroplast survival and integration efficiency, how to make them last longer and get into more cells.
Okay.
Engineering both the chloroplasts and the host cells for better compatibility, maybe modifying them genetically so the immune system doesn't attack, or perhaps even creating synthetic hybrid organelles that combine features of mitochondria and chloroplasts.
Wow, synthetic organelles.
Yeah, deep future stuff. They also point to the need for extensive safety studies and eventually maybe efforts towards potential clinical translation, although that's clearly way, way down the line.
And leveraging new tech?
Absolutely. Leveraging cutting-edge technologies like CRISPR for precise genetic modifications to enhance compatibility, nanotechnology for potentially targeted delivery of chloroplasts or light harvesting molecules, synthetic biology to build artificial light harvesting systems from scratch, and maybe developing new technologies to actually deliver light deeper into human tissues, bypassing that penetration problem.
So it's looking at science fiction potentially meeting reality through massive advancements in multiple fields of future technology. What about the broader, almost aspirational implications if something like this were ever to become widespread reality? How could it change things?
Well, the paper suggests the implications would be profound and far-reaching. For nutrition, it could fundamentally redefine what optimal nutrition even means,
How so?
potentially leading to less caloric need overall if we could generate some energy from light. Introducing entirely new ways our bodies could synthesize nutrients if the transmutation ideas held any water. Enhancing our internal production of antioxidants, potentially, and maybe leading to more sustainable nutrition globally if food dependence decreased.
And for the environment?
It's framed as a potential contribution to sustainable energy use, kind of like personal solar power, reducing reliance on traditional agriculture and food production. Potentially helping with CO2 fixation, although the scale of this in individual humans is highly questionable compared to, say, forests. But overall, potentially lowering our collective carbon footprint.
These challenges are clearly enormous, maybe insurmountable with current tech. But the potential vision laid out is genuinely breathtaking to consider. It really does force you to think differently about where energy comes from, not just for you personally, but for society and our planet. And our fundamental relationship with light and the environment.
It definitely encourages a complete reevaluation of fundamental biological processes and our interaction with the external world at a cellular level. It's provocative.
Okay, so let's quickly recap this deep dive. We started with this seemingly wild science fiction idea of human photosynthesis. And we saw that a small, intriguing step towards it has actually happened in a lab, getting plant chloroplasts into animal cells,
Yeah.
even if only temporarily and with significant limitations.
And we explored the radical, often highly speculative framework from Dr. Jack Kruse that the paper incorporates heavily, which links light to basically everything, from our fundamental energy production and nutrient synthesis to the function of crucial hormonal glands like the pineal and pituitary, suggesting our light environment is absolutely key to health and performance.
We looked at the exciting theoretical possibilities if somehow this blend of biology and light harvesting could ever work in the distant future. Imagining a world with different ways to fuel our bodies and maybe even impact the planet positively.
And we also squarely faced the very real, massive scientific, biological, and ethical challenges that make this concept currently reside mostly in the realm of future potential, perhaps even permanent science fiction for some aspects.
This deep dive really highlights a fascinating tension, doesn't it? There's the concrete, verifiable excitement of cutting-edge cellular engineering breakthroughs happening now.
Like the Matsunaga study.
Exactly. Existing alongside intriguing, but often highly speculative and far less validated, theories about light's deeper role in biology, like those from the Kruse framework.
Yeah, the achievement with chloroplasts is a real step for research, a proof of principle. But the way it's interpreted in this paper and linked to that broader quantum framework requires critical thinking and a clear understanding of what has been rigorously validated by science and what, frankly, hasn't.
Absolutely. And that leaves us with a final thought for you, the listener, to take away. This paper and our conversation today really makes you pause and ask, how fundamental is light to our biology, really? Beyond the established roles we know, like making vitamin D or setting our sleep schedules, could there be deeper, more complex ways our bodies are interacting with the electromagnetic environment around us all the time?
It's a compelling question.
And if that's true, even partially, what does that mean for the choices you make about your light exposure every single day, spending time under artificial lights versus getting natural sunlight? Gives you a whole new perspective to consider, doesn't it?
It certainly encourages thinking differently about the subtle but potentially powerful environmental factors influencing our health and energy every day.
That wraps up our deep dive into the world of human photosynthesis and quantum biology based on this research analysis. We hope you found some truly valuable nuggets to add to your understanding.